your slaves. Don't tread on us. You crossed the line. Soon you will pay. You're gonna pay for all your crimes. Don't tread on us.
In a couple of years, the Commonwealth will begin one of the biggest ecological interventions in our history, using herpes to get rid of the problem fish. But researchers are worried about the side effects. Biological controls have been used in Australia before. Some have been successful. Prickly pear, for example, was almost completely wiped out by an introduced moth. But there have also been massive failures. The cane toad is infamous for the carnage it caused after it was brought in to stop the sugarcane beetle. Bitty Bush 2 aimed to stop sand dune erosion, but destroyed native plants and habitats. Nicola Gage reports on the plans for Carpageddon.
It is potentially a massive problem that people haven't thought through. And so we're suggesting that a virus is probably not the right way to go.
Fisherman Gary Harrising has questioned whether removing so many dead fish is realistic. He says the process can't be rushed and wants other removal options to also be explored.
The original proposal about using genetics over a longer time frame was a smart move.
This kind of project on such a large scale is unprecedented in Australia. A string of government approvals will be needed, as well as support from the public. And there's a possibility that communities along the river will be tasked to help with the clean-up. There's also the question of removing potentially millions of tonnes of rotting fish. How that will happen is still being investigated.
There's a lot of things we need to determine. But so far my research does indicate that there will be huge side effects for the rest of the ecosystem.
Small changes in oxygen levels can potentially have big ramifications for native wildlife. So University of Adelaide researchers have been putting dead carp into 800-litre tubs of water to try to measure the amount of oxygen the decomposing fish use up. Although the work is in its early stages, it's showing some dramatic results.
We found that at 20 degrees, one carp can almost completely remove oxygen from the water in less than 48 hours.
Probably going to need to have some sort of complementary broad-scale control mechanism to work with it, to take full advantage of the virus. For a long time we thought that complementary approach might be so-called daughterless carp technology. Whether that occurs or not, we're not sure now, but
initially, it was a partnership between the Murray-Darling Basin Authority and the CRC allocating funds to the CSIRO to develop genetic modifications which would bias the sex ratio and control the reproductive potential of carp. And that strategy, the modelling suggests, may be effective at controlling carp in the long term, but it was a really long period until we would see the outcome of that control.
If the risks that are borne out of the legislative approval process and the public consultation process can't be resolved, clearly this won't go ahead.
Australia releases plans for its so-called Carpageddon. The Australian government has committed 15 million Australian dollars of its federal budget towards eradicating its biggest freshwater pest, the common carp. Australia's carp population has gotten so out of control that the invasive species makes up 80% of the fish biomass in Australia's Murray-Darling Basin. Adult carp, as they have no natural predators in Australia and can adapt to different environments, easily outcompete native fish species for food and resources. As current population control measures such as trapping and commercial fishing have had little effect on carp populations over large areas, the government has had to get a little more creative. To combat the rising carp population, Australia plans to release a carp-specific herpes virus into the basin. The virus attaches to their skin, gills, and kidneys and makes it difficult for the fish to breathe. The virus multiplies in the carp for about a week, and fish usually die 24 hours after they first show signs of the virus. While the virus kills the majority of carp populations, it has no effect on native fish species or other aquatic animals. Much of the $15 million the government has set aside for the project will go towards cleaning up the dead carp.
The big thing is the carp virus, and that's what we're looking at. Obviously there's a lot of questions around it. Firstly, what is it?
Well, so we've got a naturally occurring virus that was first found in Israel in the late 1990s. It's now found in over 33 countries around the world. And in those countries where this virus is present, it's caused outbreaks that have decimated their carp populations. So we've seen mortality levels of 70 and up to 100%. So this virus is extremely effective in killing carp. So as I say, it's a naturally occurring virus. It's a herpes virus, and I guess one of the characteristics of herpes viruses is they tend to be very specific to a single species. So we expected that this virus was going to be safe in the sense that it's really effective in killing carp, but safe for species other than carp. But nonetheless, the CSIRO have been doing research over the last 8 years, funded through the Invasive Animals Cooperative Research Centre, to really be doubly sure that this virus will not affect other species. So they've tested 13 species of fish, and there's some of the more iconic species like golden perch, Murray cod, but also some of the really important small species that those species feed on, like galaxids and Australian smelt. What they did is they bathed them with the virus, they injected them with the virus, and really what they were trying to do is determine whether or not they will develop the disease this virus causes. They also tested mammals, birds, amphibians, reptiles, crustacea, and what they found was that this virus will only replicate in common carp. It will only cause disease in common carp. So that's a fantastic attribute for a biological control agent. So now we know that Australian carp present in Australia, are very susceptible to the virus, and that other species won't develop the disease, which is great.
There are a number of strategies being looked at to control carp, and the one that I'm focusing on is the use of a virus called cyprinid herpes virus 3, sometimes called koi herpes virus, as a potential biological control agent for the fish. The virus has been known for since the late 90s around the world. It's not present in Australia at present, nor in New Zealand. Where it does occur around the world, it causes massive devastation in carp populations. Some people claim 100% mortality in some populations. We've done some work in the high security facility down in Geelong with this virus, and it certainly kills Australian carp. So there's no problem with the virus in that regard. Carp are certainly listed by the International Union for the Conservation of Nature as one of the most invasive of all fish species. Opinion polls taken in Australia of public opinion constantly indicate that carp are regarded right up there with cats and rabbits and toads as a major invasive species. Well, we haven't actually finally decided on how the virus will be released in a strategic manner. Technically, we'll probably release it by infecting a number of fish and releasing them into a carp population and let the fish spread the virus. The virus we know will cause high mortality. In order to take full advantage of the virus, we're probably going to need to have some sort of complementary broad-scale control mechanism to work with it, to take full advantage of the virus. For a long time we thought that complementary approach might be so-called daughterless carp technology. Whether that occurs or not, we're not sure now, but if the daughterless carp's not available, then we'll be relying on modelling approaches to help us determine the best way to make optimal use of our virus. People do worry about the potential effect on humans. We've got a number of lines of evidence that suggest that there's no basis for that belief. For example, we've actually put the virus into mice, which are a mammal like us, like humans, and we've found absolutely no evidence for disease or for multiplication of the virus. Another piece of evidence is that this virus has been causing devastation in many carp farms all around the world, and there's never been any evidence that people who work on those farms have had any sort of disease associated with the virus. We've got a couple of viruses in Australia that are quite closely related to the carp virus, and they've been here for a long time. Never has there been any evidence that they're associated with disease in humans either. And the last piece of evidence is provided by people in Europe. There was a report written for the European Commission at the turn of the century, year 2000, in which they stated that no fish virus has ever been shown to cause disease in a human. So we think that the combination of all those pieces of information are sufficient to allay any doubt about causing problems in humans. In terms of other species, we're concerned, of course, that the virus doesn't cause any problems in native fish, in amphibians, reptiles, other mammals that might share the water with infected carp. And we've done a lot of work over the last 5 to 6 years looking at the susceptibility, particularly of fish, and found absolutely no evidence of disease or indeed of the virus multiplying in 13 native fish species. No evidence of it causing problems in chickens representing birds or mice representing mammals, and we're currently looking at the effect of the virus on amphibians and reptiles, and at this stage we're about halfway through the experiment and there's been absolutely no indication so far of the virus causing any problems.
Now we're done with your tricks. Don't tread on us. Don't tread on us. We're not afraid. You work for us. 'Cause you work for us. We're not your slaves. We're not your slaves. Don't tread on us. Don't tread on us. You crossed the line. You crossed the line. So you will pay. So you will pay for all your slave. Don't tread